Baro means pressure or stretch. So baroreceptors are special nerve cells or receptors that sense blood pressure by the way that the walls of the blood vessels stretch. That information is sent from the baroreceptors to the brain to help keep blood pressure balanced. So baroreceptors are actually groups of nerve endings found within the blood vessel walls, and they can be classified into two types based on their location: the arterial ones and the cardiopulmonary ones. The arterial baroreceptors can be found on the wall of the aortic arch as well as on the wall of the carotid sinus, which is basically a bulge of the internal carotid artery just above its split from the common carotid artery in the neck. In the aortic arch, these nerve endings join up to form the vagus, or 10th cranial nerve, and in the carotid sinus they form the glossopharyngeal, or 9th cranial nerve. Both of these cranial nerves travel up towards the brainstem, carrying information about the stretch they sense in the arteries. These guys synapse at the nucleus tractus solitarius, in the medulla oblongata of the brainstem, which then relays the information to the cardiovascular centers. The cardiovascular centers are areas in the lower one-third of the pons and medulla oblongata of the brainstem, and they're responsible for the autonomic or involuntary control of the cardiac and vascular function. They do that by coordinating the sympathetic and parasympathetic branches of the autonomic nervous system. There are two main cardiovascular centers. The first is the vasomotor control center, which controls the diameter of the blood vessels, using the sympathetic nerve fibers to cause vasoconstriction. The second is the cardiac control center, which is further divided into the cardiac accelerator and the cardiac decelerator centers. The cardiac accelerator center speeds up the heart rate and increases cardiac contractility through the sympathetic outflow tract, while the cardiac decelerator center slows down the heart rate through the parasympathetic outflow tract. Notice that both the sympathetic and parasympathetic systems affect the heart rate, but only the sympathetic system has an effect on the diameter of the blood vessels, as well as the contractility of the heart muscle. This whole process is known as the baroreceptor reflex, or baroreflex in short, and takes place in seconds to minutes, allowing us to rapidly adjust our blood pressure. Alright, so as blood pulses through the carotid sinus and the aortic arch, the arterial walls get stretched out, and in response, the baroreceptors start firing more nerve impulses up to those cardiovascular centers. The higher the pressure, the higher the frequency of nerve impulses. So let's say you're running to catch the bus, and your blood pressure rises. The increased pressure stretches the walls of the aortic arch and the carotid sinus, and the baroreceptors start firing at an increased frequency. The glossopharyngeal and vagus nerve carry that increased signal to the cardiovascular centers of the brainstem. To bring the pressure back down to normal, these centers inhibit the sympathetic and stimulate the parasympathetic nervous systems. Specifically, the vasomotor center decreases the vasoconstrictive effect of the sympathetic nervous system. In other words, the arterioles dilate, decreasing total peripheral arterial resistance, and there's decreased constriction of veins, which allows blood to pool in the periphery rather than returning to the heart. Decreased venous return means that there's less preload, or less diastolic filling of the heart, and that also decreases cardiac output. Meanwhile, remember that the cardiac accelerator center is also inhibited, reducing the sympathetic effect on the heart, and letting the heart work slower and less forcefully. In other words, decreasing the heart rate and contractility, while the cardiac decelerator center is activated, boosting the parasympathetic effects on the heart, which again slows down the heart rate. Combined, these effects result in a decreased cardiac output. Since blood pressure roughly equals cardiac output times total peripheral resistance, the decrease in cardiac output and the decreasing total peripheral resistance means that the blood pressure will decrease back down to normal as well. Hopefully by that point you've made the bus. On the flip side, let's say that you're in a terrible traumatic accident and start losing a lot of blood, causing your blood pressure to fall. The decreased pressure causes the walls of the aortic arch and carotid sinus to become less stretched, and the baroreceptors start firing less frequently. The glossopharyngeal and vagus nerve carry that decreased signal to the cardiovascular centers of the brainstem. To bring the pressure back up to normal, these centers stimulate the sympathetic and inhibit the parasympathetic nervous systems. Specifically, the vasomotor center increases the vasoconstrictive effect of the sympathetic nervous system. In other words, the arterioles narrow, increasing total peripheral arterial resistance. And there's increased constriction of veins, which returns more blood to the heart rather than allowing it to pool in the periphery. Increased venous return means that there's more preload, and that also increases cardiac output. Meanwhile, remember that the cardiac accelerator center is also stimulated, increasing the sympathetic effect on the heart, and letting the heart work faster and more forcefully, meaning that it increases the heart rate and contractility. At the same time, the cardiac decelerator center is deactivated, which reduces the parasympathetic effect on the heart, which again speeds up the heart rate. Combined, these effects result in an increased cardiac output, as well as an increase in total peripheral resistance, which raises the blood pressure back to normal. In this case, these changes can save your life. Now, the other type of baroreceptors are called cardiopulmonary baroreceptors, and they're embedded within the walls of the right atrium, right ventricle, and pulmonary artery and veins. These are all relatively low pressure areas, so the stretch of the walls mainly depends on the blood volume flowing through, or the fullness of these vessels. This is why these baroreceptors are mainly responsible for the regulation of blood volume, and are also known as low pressure or volume baroreceptors. If the blood volume increases, the cardiopulmonary baroreceptors start firing more frequently through the vagus nerve toward the cardiovascular centers of the brainstem. These centers then send signals toward the heart, increasing its rate and thus the cardiac output. Increased cardiac output means more blood reaches the kidneys, so more water and sodium can get excreted in an attempt to lower blood volume. This is called the Bainbridge reflex. In addition, when cardiopulmonary baroreceptors sense high blood volume, they also send their signal via the vagus nerve to the hypothalamus of the brain, telling it to reduce the production of vasopressin or antidiuretic hormone. This results in decreased water reabsorption from the kidney, letting more water get lost in the urine. Cardiopulmonary baroreceptors also stimulate the secretion of atrial natriuretic peptide, which comes from the atrial muscle cells. ANP causes renal arterioles to dilate, allowing them to receive and filter even more blood, while also inhibiting water and sodium reabsorption. On the other hand, if the volume of blood decreases, cardiopulmonary baroreceptors get less stretched, which reverses all of the effects. The heart rate slows down, more antidiuretic hormone is made, while less atrial natriuretic peptide is made. All this results in less water and sodium excretion, which helps restore blood volume. Alright, as a quick recap. Arterial baroreceptors are located in the aortic arch and carotid sinus, and respond to blood pressure changes, sending information through the glossopharyngeal and vagus nerves to the cardiovascular centers in the pons and medulla. These, in turn, through the autonomic nervous system, modify total peripheral resistance and cardiac output. Cardiopulmonary baroreceptors are found in the low pressure regions of the heart and the pulmonary vessels, and regulate blood volume by influencing water excretion by the kidneys.
Chemo refers to the chemical composition of the blood. So chemoreceptors are the special nerve cells, or receptors, that sense changes in the chemical composition of the blood. That information is sent from the chemoreceptors to the brain to help keep the cardiovascular and respiratory systems balanced. Alright, according to their location, chemoreceptors can be classified into two types, peripheral and central ones. Now, the peripheral chemoreceptors are so named because they live outside the brain. They are actually tiny bodies, or clusters of nerve cells, and they include the aortic body, which sits along the wall of the aortic arch, and the carotid body, which is located at the point where each common carotid artery splits into the internal and external carotid arteries, running alongside the neck. Both the aortic and carotid bodies are bathed in arterial blood, and they carefully monitor changes in the concentration or partial pressure of oxygen, PO2 for short, but also the partial pressure of carbon dioxide, or PCO2 for short, as well as the concentration of hydrogen ions, which determines blood pH. The aortic body sends this information along to the vagus, or 10th cranial nerve, and the carotid body sends this information along to the glossopharyngeal, or 9th cranial nerve. These two large nerves travel up towards the respiratory centres, which are in the brainstem. The respiratory centres are groups of neurons located in the pons and the medulla oblongata, and they're responsible for the autonomic or involuntary control of breathing. The respiratory centres also communicate with the cardiovascular centres. The cardiovascular centres are areas in the lower one-third of the pons and medulla oblongata of the brainstem, responsible for autonomic or involuntary control of the cardiac and vascular function. They do that by coordinating the sympathetic and parasympathetic branches of the autonomic nervous system. Now, there are two main cardiovascular centres. The first is the vasomotor control centre, which controls the diameter of blood vessels using sympathetic nerve fibres to cause vasoconstriction. The second is the cardiac control centre, which is further divided into the cardiac accelerator and the cardiac decelerator centres. The cardiac accelerator centre speeds up the heart rate and increases the cardiac contractility through the sympathetic outflow tract, while the cardiac decelerator centre slows down the heart rate through the parasympathetic outflow tract. Notice that both the sympathetic and parasympathetic system affect the heart rate, but only the sympathetic system has an effect on the diameter of the blood vessels and the contractility of the heart muscle. Okay, so let's say that you have hypoxemia, meaning a decrease in the arterial partial pressure of oxygen, PO2, along with hypercapnia, an increase in the arterial partial pressure of carbon dioxide, PCO2, and acidemia, a decrease in blood pH. As blood pulses through the aortic arch and the common carotid, these changes get detected by the aortic and carotid bodies, which start firing more nerve impulses up through the vagus and glossopharyngeal nerves towards the cardiovascular centres of the brainstem. In response, the vasomotor centre increases the vasoconstrictive effect of the sympathetic nervous system, causing arterioles to narrow, raising the total peripheral arterial resistance. In addition, the vasomotor centre limits blood flow to the peripheral organs, like the muscles, to help conserve oxygen for the brain and heart. It also decreases the metabolic rate to help reduce carbon dioxide production. There's also increased constriction of veins, which returns more blood to the heart rather than allowing it to pool in the periphery. Increased venous return means there's more preload, and that also increases cardiac output. Now, another effect is that the cardiac accelerator centre gets inhibited, reducing the sympathetic effect on the heart, so that it can work slower and less forcefully. In other words, decreasing the heart rate and contractility. While the cardiac decelerator centre is activated, boosting the parasympathetic effects of the heart, which again slows the heart rate. This last bit might sound a bit counterintuitive, but stay tuned. Now, it turns out that peripheral chemoreceptors also send impulses through the vagus and glossopharyngeal nerves, which reaches the respiratory centres in the brainstem. And they cause you to suck in more oxygen and blow off more carbon dioxide by getting the diaphragm and the chest wall muscles to breathe faster and deeper. Now, this causes airways to get stretched, and that's detected by another type of receptor called the pulmonary stretch receptor. And these are located in the smooth muscles lining the trachea and central bronchi. Once stretched, these receptors start firing through the vagus nerve back to the pons and medulla, where they tell the respiratory centres to slow down. At the same time, though, they stimulate the cardiac accelerator centre, and so the sympathetic effect on the heart is increased, and this makes the heart rate and the contractility increase. All the while, the cardiac decelerator centre is inhibited, and thus the parasympathetic effect on the heart, which again helps the heart rate to speed up. In other words, they reverse the direct effect that the peripheral chemoreceptors had on the cardiac centres. Ultimately, this makes the heart work faster and stronger, letting the cardiac output, CO, and blood pressure, BP, rise. This is essential to push more blood to the lungs so that it can get supplied with oxygen and freed from excess carbon dioxide. Now, peripheral chemoreceptors work in concert with the central chemoreceptors, which take their name from the fact that they're part of the central nervous system, particularly the brain. Central chemoreceptors are clusters of neurons packed together in the ventral or frontal surface of the medulla oblongata at the base of the brain, right next to the respiratory centres themselves. The central chemoreceptors are bathed in cerebrospinal fluid, and they sample the fluid to assess for changes in the partial pressure of carbon dioxide, PCO2, and pH. That's different from the peripheral chemoreceptors, which are primarily sensitive to changes in the partial pressure of oxygen, or PO2. When the partial pressure of carbon dioxide rises in the blood, carbon dioxide molecules rapidly diffuse across the blood-brain barrier and build up in the cerebrospinal fluid. Cerebrospinal fluid is mainly composed of water, so carbon dioxide binds to water molecules to form carbonic acid, H2CO3, and that rapidly breaks down into hydrogen ions and bicarbonate ions. As hydrogen ions build up too, the pH of the cerebrospinal fluid starts to drop. So both the increased partial pressure of carbon dioxide and the decreased pH together trigger the central chemoreceptors to stimulate the nearby respiratory centers to increase the respiratory drive. Now, what makes the central chemoreceptors special is that, due to their location, they are also influenced by changes in the blood flow to the brain. Imagine, for example, that there's a large mass inside the head which raises the intracranial pressure, squashing the blood vessels heading in and out of the brain and causing brain ischemia. This means that all of the carbon dioxide that gets released by the brain cells as a byproduct of their metabolic processes can't escape through blood flow. So inevitably it starts accumulating in the cerebrospinal fluid, or CSF, also decreasing its pH. Once again, this gets detected by the central chemoreceptors, which, via the vasomotor center, leads to an arteriolar and venous constriction, redirecting more blood flow to the brain. That increases the total peripheral resistance, TPR, which raises the blood pressure, BP, helping the blood pressure to stay higher than the cerebrospinal fluid pressure so that blood can continue to flow to the brain. This increase in blood pressure in response to increased intracranial pressure is called the Cushing reaction, named after the neurosurgeon Harvey Cushing. Alright, so as a quick recap, peripheral chemoreceptors include the aortic and carotid bodies, which respond primarily to decreases in the partial pressure of oxygen, PO2, and secondarily to increases in the partial pressure of carbon dioxide, PCO2, and decreases in blood pH, sending information through the glossopharyngeal and vagus nerves to the vasomotor and cardiac centers of the pons and medulla. These, in turn, through sympathetic stimulation, increase total peripheral resistance and cardiac output, raising blood pressure. Central chemoreceptors are found in the medulla and are more sensitive to increases in the partial pressure of carbon dioxide and decreases in pH, which, again, through the vasomotor center, stimulates sympathetic vasoconstriction, raising blood pressure.
The main job of the cardiovascular system is to keep the blood moving, and to help facilitate that, blood pressure and, more importantly, blood volume is kept under tight control. A major way the body does that is through a set of hormones that make up the renin-angiotensin-aldosterone system. But first things first, everything starts in the kidney. Now, within each kidney, blood from the renal artery flows into smaller and smaller arteries, eventually reaching the tiniest of arterioles called the afferent arterioles. After the afferent arteriole, blood moves into a tiny capillary bed called the glomerulus. The glomerulus is part of the functional unit of the kidney, called a nephron. There's about 1 million nephrons in each kidney, and each of them consists of a renal corpuscle, made up of the glomerulus and the Bowman's capsule surrounding it, and a renal tubule. The renal corpuscle is where blood filtration starts. Interestingly, once the blood leaves the glomerulus, it does not enter into venules. Instead, the glomerulus funnels blood into efferent arterioles, which divide into capillaries a second time. These capillaries are called peritubular capillaries, because they're arranged around the renal tubule. Now, the renal tubule is made up of a proximal convoluted tubule, the nephron loop, also known as the loop of Henle, which has an ascending and descending limb, and finally the distal convoluted tubule. As filtration makes its way through the renal tubule, waste and molecules like ions and water are exchanged between the tubule until finally urine is formed. At the same time, the peritubular capillaries reunite to form larger and larger venous vessels. The veins follow the path of the arteries, but in reverse, so they keep uniting until they finally form the large renal vein, which exits the kidney and drains into the inferior vena cava. Alright, now if we zoom into the wall of the afferent arterioles, we'll find a very special kind of smooth muscle cell, called juxtaglomerular cells, because they're next to or juxta the glomerulus. The main job of these cells is to always keep an eye open for signals that the blood pressure and or blood volume needs to rise. These signals can come in three ways. First, juxtaglomerular cells are mechanoreceptors, also called baroreceptors, and they're designed to mechanically feel if there's low blood pressure in the incoming blood. When they're stretched by an increased blood pressure, they inhibit renin release. When they're collapsed from low blood pressure, they stimulate renin release. Second, juxtaglomerular cells are supplied by sympathetic nerve fibers. The sympathetic nervous system is activated by mechanoreceptors stationed strategically in the aortic arch and carotid sinus to measure the immediate blood pressure coming out of the heart. If they're stretched, then the sympathetic nervous system will be downregulated. However, if they collapse secondary to low blood pressure, then the sympathetic nervous system is activated. Specifically, the sympathetic nerves stimulate the B1 adrenergic receptors on the JG cells to stimulate renin. The third signal for juxtaglomerular cells comes from specialized cells in the wall of the distal convoluted tubule called macula densa cells. Macula densa cells are chemoreceptors that can sense when glomerular filtration rate increases or decreases based on the quantity of sodium and chloride ions flowing through the tubule. Here's how it works. When blood pressure rises, renal blood flow and, as a consequence, glomerular filtration rate also increases. This means that there's more fluid and more dissolved sodium and chloride ions that reach the macula densa. Now, if the opposite happens, and there's decreased fluid and sodium and chloride ions getting to the macula densa cells, then that sends a signal to the juxtaglomerular cells in the afferent arteriole. The major signal communicating between the macula densa and the juxtaglomerular cells is prostaglandins, especially PGE2. As a result, the use of NSAIDs, or non-steroidal anti-inflammatory drugs, can block this signal and impair the response of the kidney to reduce blood pressure. All three types of signals stimulate the juxtaglomerular cells to secrete renin and initiate the renin-angiotensin-aldosterone system pathway. Renin is an enzyme that gets into the plasma and looks for its primary substrate, angiotensinogen. Angiotensinogen is a large protein made up of over 400 amino acids that's produced by the liver and is always hanging out in the blood. When they meet up, renin cleaves off a huge chunk of the angiotensinogen protein, leaving behind a tiny fragment called angiotensin 1 that's just 10 amino acids long, kind of like a tiny pearl found within a giant oyster. Angiotensin 1 floats through the blood until it reaches various capillaries in the body. Now, endothelial cells in general, but mostly those lining the vessels in the lungs, have an enzyme on their surface called angiotensin-converting enzyme, or ACE for short. Angiotensin-converting enzyme grabs angiotensin 1 and chops off 2 of its amino acids, leaving behind an 8 amino acid chain that's a really powerful hormone, called angiotensin 2. Angio means blood vessel, and tensin means making everything more tense. So angiotensin 2 goes to the smooth muscles lining arterioles all over the body that contain the angiotensin 2 receptor, and causes them to tense up or contract, which leads to vasoconstriction of those blood vessels, and increases resistance to blood flow. So angiotensin 2 travels through the blood, and when it reaches the kidneys, it binds to angiotensin 2 receptors predominantly located along the efferent arterioles. Therefore, the efferent arterioles are much more responsive to angiotensin 2 than the afferent arterioles. So when there are low levels of angiotensin 2, only the efferent arterioles constrict, and this makes less blood leave the glomerulus, or said differently, it makes more blood remain in the glomerulus, thereby increasing intraglomerular pressure and preserving the glomerular filtration rate. However, when there are high levels of angiotensin 2, both the afferent and efferent arterioles constrict, and this decreases both renal blood flow and glomerular filtration rate. Angiotensin 2 also acts on the cells that line the proximal convoluted tubule, making them reabsorb more sodium ions from the filtrate. And by the process of osmosis, water starts to follow the sodium ions. Angiotensin 2 also acts on the hypothalamus, which is at the base of the brain, where it stimulates thirst and increases the production of antidiuretic hormone, or ADH. ADH has its main effect on the cells lining the distal convoluted tubule and collecting duct, where it stimulates more aquaporins to get into the luminal surface of the cells. Aquaporins make these cells more permeable to water, increasing water reabsorption. Ultimately, the increased thirst and water reabsorption increases the total circulating blood volume, or blood flow, symbolized as Q. In addition, the increased peripheral arterial resistance increases the total body resistance, symbolized as R. Since the change in blood pressure P equals blood flow Q times resistance R, the blood pressure rises. But angiotensin II's work isn't over yet. It also stimulates the adrenal glands, which are a pair of glands that sit on top of each kidney. If we slice an adrenal gland open and zoom in, we'll see that it's made up of two parts, an inner medulla and an outer cortex. The cortex is further subdivided into three more layers: the zona glomerulosa, the zona fasciculata, and the zona reticularis. Angiotensin II stimulates cells in the outermost layer, the zona glomerulosa, to secrete aldosterone. Aldosterone belongs to a class of steroids, or lipid-soluble hormones, called mineralocorticoids, which help regulate the sodium balance of the body. Aldosterone does that by acting on the cells that line the distal tubules and collecting ducts. Aldosterone gets into those cells and binds its receptor, forming an aldosterone receptor complex. This complex is able to control the expression of genes, increasing levels of some proteins over others. One protein that gets expressed a lot is the sodium-potassium ATPase ion pump, which sits on the basolateral surface of the cells. These pumps push sodium into the blood and pull potassium into the cells. Ultimately, this helps drive a concentration gradient that pulls sodium and water out of the lumen of the tubule and into the blood, and secrete potassium. In fact, most patients with intense activation of the renin-angiotensin-aldosterone system can be profoundly hypokalemic. As before, this helps to boost the blood pressure. Keep in mind, though, that this process can take several hours or even days, so it's considered a late response to the renin-angiotensin-aldosterone system. Alright, as a quick recap. Whenever there's a decrease in blood pressure, as detected by the baroreceptors of the carotid sinus, or aortic arch, or the juxtaglomerular cells, the sympathetic nerves getting stimulated, or the macular densa cells sensing less sodium and chloride ions flowing through the tubules, kidneys secrete renin that converts angiotensinogen to angiotensin 1, and then angiotensin converting enzyme converts angiotensin 1 to angiotensin 2. Angiotensin 2 causes the efferent arteriole to constrict more than the afferent arteriole, which increases the glomerular filtration rate. It also causes the proximal tubule to reabsorb more sodium ions from the filtrate, increases thirst, and helps increase blood pressure, while it also stimulates the adrenal cortex to release aldosterone, which gets the kidneys to retain sodium and water.